Hello, everyone. Good morning. So welcome to the second lecture of Project Sense Seminar of Mobile Genomics. So for today, we will go to Introduction to Sequencing. And the previous weeks, we already assigned projects to each of each of the students. And I hope these projects going well. So I hope you are meeting with the mentors in every week. So we will have a follow-up meeting probably in every week. So you will meet with the mentors as we discussed, and these mentors will provide helpful feedback, comments to each of the students based on the assigned project. Okay, so for today, I think we will cover the topics related to sequencing, DNA sequencing. So for now, we have three main topics for this lecture. The first, what is genome analysis? And second, what is intelligent genome analysis? So we'll define genome analysis in general, and then we show the need, why we need really intelligent genome analysis. And at the end, we will show how to analyze the genome, basically, or how to sequence the genome, read it first, and then try to get useful information out of this. In the following weeks, we will continue with genome analysis and show the method used to build or to do read mapping or to build these the complete genome out of these small pieces. So let's for now start with the first topic. What is genome analysis first? So if we talk about data analysis in general, so I like this saying from Richard Hamming, that the purpose of computing is to gain insight, not numbers. So when you want to do some computation, you are not looking for raw numbers, but rather to interpret these numbers and get useful information out of it. And the same thing with genome analysis. So given that there are a large number of people living on Earth and maybe other planets, so we want to know out of curiosity, or it could be some other reason, scientific reason, what cause all these differences between individuals. So you can see there are a lot of phenotypes, traits between these individuals in the picture. With that, genome analysis is basically to analyze or identify, measure, compare genomic features such as DNA sequences. Not only comparing the DNA sequences, but also to know the functional element annotation, what each sequence in the DNA causes, for example. And we want to do this at genomic scale. So that we want to analyze the entire genome and see what's the function of every and each piece in this DNA sequence. Okay, so how we do this genome analysis. So the first step is to take a sample out of the human body, for example, and then you can buy one of these commercialized kits from market or from pharmacy, and then you can take that swab, send it by post to the company, and the company will do the rest of the process on their own. So at the company side, what they will see is these chromosomes after, of course, some wet lab processes, such as, for example, library preparation, where they isolate the content of the sample, for example, red blood cells and plasma and maybe other content in the sample. They will extract the genetic material only and then start to sequence these genetic material. So we have in human genome these chromosomes. So our goal is to read the content of these chromosomes within a cell or out of each cell. So, you know, the chromosome, part of it coming from the mom, the other part coming from the dad. So it's inherited material. But there are also some genetic materials are coming from nowhere, not from the mother, not from the father, but due to some reasons, during cell replication, there might alteration happening to the cell that cause de novo genetic mutations, for example. When we look deeper to these chromosomes, we find that the structure of the DNA is double helix structure, where adenine is chemically binded to thymine, and then cytosine is chemically binded to guanine, and so on. And these chemical bonds are like really strong, and it will be difficult for us to read the content without splitting the double helix structure into two strands of DNA, and then start to do some processes on these DNA strand, as we will explain toward the end of the lecture. Okay, assume now we got the DNA sequence or the sequence out of this process when we want to get the chromosomes, what we can get out of it. This is an example. Assume we have the DNA sequence for individual number 4 over here. We have also other DNA sequences for other individuals. And we have also some health information such as blood pressure. And now, because all of you are smart people, I ask you what may cause or what is the genetic cause for blood pressure. Now, you may guess or you may correlate these genomic sequences to the value of blood pressure. If you look at SNP2, you can see when the blood pressure is really high, 180, it is G at SNP2 location. When it's also low, like 105, it's also G at SNP2. So you cannot make any correlation between the SNP2 and the blood pressure values. But when you look to SNP1, you see it's really correlated such that whenever we have a T at that location, the individuals have low blood pressure. But when we have a C at that same location, SNP1 location, then that is correlated with high blood pressure. So you can make some correlation based on this correlation and some historical medical data. You know that if you have this content in your genomic sequences or in your genetic material, then probably you have this disease. And also based on the historical data, you know that the previous patient who have this disease also getting better because they got this treatment or this kind of medication and so on. So you can leverage all this information together to prescribe a very good medication course for the patient based on this information. So that is what we call Genome-Wide Association Study, which is first to try to detect the genetic variation that may cause some of the disease. For that, we need 2 groups of people. One of them, the control, those people who may not have the disease, the target disease. And we have cases where we know that they may have this disease or they already have it. And then we get large sample, large groups. So both of these groups are really large so that we can remove all noises, for example. And we build what we call a Manhattan plot. For example, this, and you can guess why we call it Manhattan plot. So we know all the variation or all the genetic material for all these individuals, and then we try to plot the frequency of each of these genomic variation at each and every location. So the x-axis here represents the chromosomes, and we represented each location the frequency of every genomic base or genomic character. And when a certain threshold exceeded, then we know that there are a lot of these bases or a lot of these individuals are caused by certain variation. And this variation or any frequency of this variation exceeding the threshold might represent the cause for that disease. Now, this could be a single variation or it could be multiple variation at multiple locations and spread it over all chromosomes. And yeah, and you can see all these snaps exceeding the higher frequency or the frequency threshold, as I mentioned, in cases than control. Now, there are other studies that done with large groups of people, such as transcriptome-wide association studies. So I suggest everyone to read this paper if you are interested in knowing more about other studies. And these studies are really important and really used even in clinical setup, where one of these databases is called OpenSNP, that accommodates a large number of information, genetic information, and as well as health information about individuals. Of course, these individuals are anonymized. You don't know who they are, but basically you know about their diseases and the treatment they went through while they're having these diseases. So this example showing you at chromosome 19, at this location in chromosome 19, if you have one of the genetic variation to be T, for example, then you have this probability to get better or to respond to treatment if you have hepatitis C. But if you have C at that location, then 80% of hepatitis C patients also respond to treatment. But then if you have a C and T in that location, one coming from the mother, one coming from the father, then the probability of getting better or responding to treatment become less than if you have C and C, for example. And based on this information, you can do personalized medicine or prescribe some medication or some treatment that is suited or well suited to individual based on their genetic sequences. Now, the personalized medicine becomes the norm these days. So in critically ill infants, for example, when they are admitted to hospital and you have very short time to respond to their illness, you have to take action, quick action before they die. And for that, if you don't know what are these symptoms and you cannot map a disease to these symptoms, probably the best way is to do whole genome sequencing. So you need to read their DNA first and then analyze the DNA, map it to the disease that you already know in the database, and map all these content of the DNA to the disease, and you may figure out some genetic causes for such illness. And based on that, that help you a lot to avoid morbidity, reduces hospital stay length by 6% to 69%, reduces also the inpatient cost, which significantly can save the taxpayer money. This has become really a typical day in the UK. Whenever you have seriously ill children admitted to hospital, then the first thing they do is whole genome sequencing, starting from 3 years back. If you think that disease might be caused by a single or few variation genomic, that is not true. There are really large structural variation. For example, autism caused by a deletion of 500,000 bases in chromosome number 16. Same thing with obesity, underweight, and so on. So for example, here, look at the obesity, deletion of 500,000 bases from chromosome 16, but underweight is totally the opposite. Instead of deletion, we have a duplication of 500,000 bases. And these are the paper. You can read more about each of these diseases and the analysis they did to figure out the cause for such challenging diseases. I also recommend everyone to read this paper. So now we have 2 recommended readings for today. We have more in the following slides. These are really very good paper. I would say one of the best in the recent years. So I recommend everyone to check this paper where you got a nice review about all type of structural variation that may cause disease, and you get more information about the computational algorithms used to figure out these variation. Okay, that was all for the first section. So now let's go to the second topic, which is about intelligent genome analysis. So what is intelligent genome analysis? So first, we want these analyses to be really fast. As I show in the previous slide, there might be life-threatening causes to the health, and we would like to figure out what caused these variants or these causes. And for that, you want to quickly take an action to stop the illness or provide a treatment to the ill children, or it could be just grown-up adults and so on. So that is about the bandwidth, how fast you can do single genome analysis. But also, when we are talking about a typical day or a routine process in the hospital, then the energy efficiency become a concern here. You can adapt all these sequencing machine plus the computer used for the analyses and so on. So we are talking here about intelligent architectures as well that may provide more efficient more energy efficient architecture or machines plus reducing the latency, because you could do hardware acceleration to reduce the time needed to do single analysis compared to software optimization, for example. But remember also, the DNA is a valuable asset, so you don't want to release it publicly on your GitHub page, for example. You don't want to put your genomic data over there, or you don't want to give it to third party, even if they are clinical senders, because they might also make it publicly available for research purposes or other purposes. This genomic material has more information than what we need, because they might reveal some secret information about your relatives, about yourself, and so on. You don't want to tell everyone that you have this disease and that disease and so on, because you might be blocked from, for example, health insurance policies and so on. And that is raising concern about the privacy, not only security. So security and privacy are really, both of them, important to the DNA sequences or genomic material in general. We talk about the importance of having very fast genome analysis, but when we talk about the entire population or entire the city scale analyses, then it's not really important how fast a single genome analysis is, but also how many genome analyses you can do in parallel at the same time within a given day, for example. That is really important, which we call scalability. So it's really important to be able to scale the analysis to a large number of people. So you have to do it, all of them, with high parallelism, rather than waiting to finish the first analysis and then the second analysis and so on. We want also to not compromise accuracy. So it's really important to avoid accelerating genome analysis while we are compromising accuracy. We don't want to provide heuristic algorithm or very fast algorithm that provides large false positive. And this kind of algorithm might be useful in some application, but in some other application might not be useful at all. So the accuracy should be maintained in all cases. Okay, now, given the definition of the intelligent genome analysis, does intelligent genome analysis really matter? Okay, you might watch already these two movies, Gattaca and Tomorrowland. So if you see or if you notice this smartwatch where you just put your finger and in mere second it can analyze your DNA and figure out who you are based on your genetic material. This is our ultimate goal, basically, to have something really portable, really accurate, and can analyze your DNA very efficiently, very quickly, using limited computational resources. We are not talking about clusters or cloud computing, because some remote areas, you don't have access to these facilities. Or in some cases, the network will be overwhelmed, as we have in COVID-19 the first year or the first 2 years. The internet network is really overwhelmed by the large number of people using it at the same time. We don't want such thing to interrupt these analyses. So just analyze on your own, and that will enable a lot of application by having these portable devices to do DNA analysis. And that is show the need for intelligent architecture. So we want to study where to do the analysis, or which part of the compute stack we want to perform analysis, and whether we want to move the data from the sequencing machine to somewhere else or perform the analyses inside the sequencing machine. We will talk more about this topic in the following lectures. And this is how we envision it in the future. This is actually a real device, but the accuracy is really low for these kind of devices. We want something really portable, very small machine. You can put the lab prepared sample over here, and then it will do sequencing, send the data to the phone to do the further compute analyses to interpret your data. This is our paper about privacy-preserving genome analysis. I recommend everyone to read it, where we show the attacks that are possible on your genomic material, plus some other side channels such as social media and some more public available information. For example, hair color, eye color, all these you can infer them from Twitter, from Facebook, and so on. You can correlate them with the genetic material and figure out and the other genetic material of your siblings and other family members. This is the paper again. And there are some DNA tests available in the market that can provide you privacy-preserving tests. So they also control what they are going to sequence or what they are going to show, and then destroy the other information or the other genetic material, so they are not publicly available for the rest of the world. Now, remember, during the COVID-19 or any of the disease outbreak, for example, here, paper show the analyses for Ebola. You want normally to have these devices in remote areas, for example, in Africa, where you don't have large facilities or you don't have internet connection, you don't have power, and so on. So that is showing you the need for portability. Portable analysis is not the only thing. You want also to do it at scale. So here, a real application for COVID-19 sequencing. So you can detect the COVID-19 positive cases based on sequencing data and not only using PCR, because as you know, PCR is a targeted analysis where you already know in advance what you are looking for, and you are only looking for a single species to exist. But with sequencing, you can get diversity, you can know more than a single species. For example, you can study the bacteria that's associated with COVID-19 or viruses and so on. You can study a lot of species by doing just single sequencing run. And you don't need to know the exact strand or the exact genomic sequence or material you are looking for. For example, here we are talking about SARS-CoV-2 viruses, but you may have in advance some closely related species, and then using sequencing, you can do metagenomic studies to figure out what you have in your sample. And then these are the main differences between sequencing-based analyses and PCR-based analysis. This is the metagenomic study. So for example, assuming you have a crowd and you want to analyze what we have in such crowded places. So you want to take a sample from these cities. It could be train stations, train door, or universities, classroom, and so on. So you take a sample and you want to analyze it. And this was a huge study in 2015, where they take about 1,500 samples from New York subway stations. Then they try to do genomic or metagenomic studies to figure out what do we have in all these stations. So there was about 50% bacteria and other 50% unknown organisms. And that is to show you that we still don't know much about all these kind of microbiome. However, in 6 years later, there was a huge study published recently about doing metagenomic analysis in 60 cities. And now you can figure out the number of samples is really much larger than before. It's about 3x, 3 to 4x more than what we have in the New York study. And you can see the importance of doing these kind of analysis everywhere, to all countries, to all sites, and even in space, where you want to figure out what kind of livings we have over there. One important thing out of the New York subway station, they find the plaque, which is Yersinia pestis. And for those who don't know what is it, there is a bacteria infection that killed more than 1/3 of the population of Europe within a few years in the 14th century. So it's really important to do these analyses regularly, such that you figure out what was harmful, what was not harmful bacteria, so you take action on time. And this was covered by the news and raised a lot of alarm in that time. This is in 2015. However, when we went with the traditional method, when they do culture to these samples, they didn't find any trace to these bacteria, and that what some people called as a failure of bioinformatics. So that is, again, is not a failure of bioinformatics, but rather it's really the failure of the tool used to do the analysis. So the accuracy of the tool wasn't really high, and that caused a lot of false positives. And that can tell you that there's a critical need not only for fast analysis, but also for accurate genome analysis. So as I said before, the speed might not be the only thing, or might not be the only important aspect in this analysis, but also to maintain very high accuracy. So to sum up, or to conclude, we want to achieve intelligent genome analysis, but we still don't know how to do it, how and where to enable fast, accurate, cheap, privacy-preserving, and exabyte scale analyses of these genomic data. That is still open question for research. And we did a lot in our research group to try to address these questions, but we're still far from achieving the end goal, which is very portable, very energy efficient analyses within small devices. Now, toward the end of this lecture, I will just go briefly through sequencing machines and show how it works, just briefly, just to give you a glimpse out of how these devices work and what kind of data we got out of it. So our ultimate goal is first to have this genetic material or genetic sample, and then we get out of it a complete sequence of our DNA. Unfortunately, we don't have these machines so far. These machines just provide you segments of your DNA. Why is that? I will leave this as an open question to all of you, but you will find the answer to this question in this paper and in the following paper as well. I recommend everyone to go through these two papers, but just to give you a hint about the reason why we, until now, with all the advancement in sequencing technology, we're still getting chunks of the DNA, basically because we are trying to isolate the content of the cell or the content of the blood or the sample you take from the genetic material. So there are many more than just DNA in the sample, and we don't want all of these things. So basically we shear it, for example, or rotate the sample in very high speed that try to isolate the plasma from the DNA and so on. And that causes alteration to the DNA molecule, so such that it's divided into pieces. These pieces can be randomly or can be organized or regular way of fragment these DNA strands, and then you add them to the sequencing machine. And then the sequencing machine will read these fragments, but not the entire molecule. And there are many other reasons discussed in the two papers, so I recommend everyone to go through them to get more information about why these sequencing machines still providing segments of the DNA rather than the complete DNA sequence. For that, genome sequencer is still a chopper, so they provide you segments of your DNA. The today sequencing machine performing really well. They can provide up to 10 to the power 12 bases in about 44 hours. That is about 2 days, and with less than $1,000. However, because of these pieces, we cannot directly interpret them. So you need what we call genome analysis step to link these pieces together, and then you can detect the variation. And there are a lot of these choppers or sequencing machines. This guy, for example, is a fridge size, really huge. This is also a huge one. This is you can put it in your bench. And these are huge with 4 to 5 GPU cards to do the analysis inside them. So this is a really huge device. But this is a portable one. It's handheld, as you can see. It's hand-sized machine called MinION from Oxford Nanopore technology. It's very cheap USB stick. It's about, it costs $1,000, I think. But also you need to add some reagents and some materials with each run. Then the cost will add up. This is also another machine from Oxford Nanopore device. It's really small, can be attached to your mobile phone. And each of these machines basically has its own properties. So here, the latest machine from Oxford Nanopore, you can see the read length they provide is really long, like 2 million bases or character. And the yield per device is really different also from one machine to another. This is the same machine inside this, but this is able to do the analysis as it has some compute infrastructure, but this is just sequencing. So you still need to move the data to a machine or a laptop to do the analysis. This has GPU to do base calling and to do other processes. That is a pre-processing step before you do the analysis and so on. You can see the price also is different based on all these properties I mentioned. When it comes to the second company called Illumina, they also have a large number of machines, and you can see the throughput, the run time is also different. But when it comes to the read length, almost all of them, they provide very short reads. That is maximum 150 base pair, and on average is 100 long sequences. The cost is really varies as well, but it's still way cheaper than what we have in Oxford Nanopore devices. This is the latest machine they have, and they have compute capabilities inside the machine. So you can do not only pre-processing as we have in Oxford Nanopore, but you can also do real-time analysis, the full genome analysis inside it on FPGA card called Dragon. Okay, now coming to Illumina machine, how the sequencing works. So we have a glass flow cell, and we have the DNA strand, and we know that in advance that adenine is chemically binded to thymine, and guanine is chemically binded to cytosine. So what we use this information to do a chemical reaction ourselves, and we watch this chemical reaction and see what will happen. So we add the thymine. We have no information about the content of this DNA strand. So we don't know if there is an A here or not. We add the thymine, and we have an optical sensor to watch the chemical reaction, which is basically a camera. So once the chemical reaction is happening between the thymine and the base, if the chemical reaction happening, then the optical sensor will take or will capture that, and it will give you the letter A. How do you know it was A? Because only adenine will chemically bind to thymine. If we add thymine and a chemical reaction happening, then we know that was A over there. So we repeat the same thing with all 4 molecules. So since we don't know if this is A, we add thymine. If the chemical reaction happened, then we are done. But if it is not happening, we wash the surface from thymine, and then we add cytosine, guanine, adenine, gradually one by one, wash surface and watch the chemical reaction. It's not happening, watch the surface, add the next molecule, and so on. We don't do this to a single DNA strand, but we do it with high parallelism. So we have many of these DNA strands attached to the glass surface. So all of them are done in the same cycle. All the sequencing to all these DNA strands is done in the same cycle, so that we can do what we call high-throughput sequencing, or highly parallel sequencing process. And this what we call as a read or DNA fragments. Now check this video, it's very interesting to check. I really enjoy watching this video. You can watch it and get more information about real sequencing machine. When it comes to second company, so the first, the things we were explaining in the previous slide were about Illumina, which producing short reads. Now this is about Oxford Nanopore, which producing very long reads. So the technology is basically have many nanopores. For example, here, this is a nanopore or graphene nanopore, and it's attached to electrical current source passing through this pore. This is the DNA strand or the target strand that we are going to sequence. There is nothing controlling it except the weight of this DNA strand and the gravity. So the gravity will let this DNA strand just glide through this pore, gliding down. Now the current will pass through the DNA strand, and the DNA over here will alter the phase or the amplitude of the input electrical current, and then the alteration will be observed by a sensor over there. And based on this alteration, we can know if this is adenine, guanine, thymine, and so on. So nanopore is a nanoscale hole, really less than 20 nanometer in diameter. And in nanopore sequencers, we use a current passes through the nanopores. When the DNA strand passes through the nanopore, the sequencer measures the change in current. This change is used to identify the bases in the strand with the help of different electrochemical structure of the different bases. So basically the hydrogen, the other atoms in each of these bases will affect the amplitude of this electrical current. Again, this is another video that I recommend everyone to watch it about the sequencing process. So I hope by that I covered the general idea on how we do sequencing in these two machines and how they work in general. And you still, as I mentioned before, the common disadvantages between all these machines is that none of them provide you any information about what read comes next after the current read. We don't know any information about their order, their location, and so on. For that, we will use this analogy as solving the puzzle. So think about, you have a lot of pieces that are reads, and you have a reference picture. Based on the reference picture, you try to match each piece to this reference picture and then solve the puzzle. Okay, now for the puzzles, when you go to the market, try to buy a puzzle, there are two options, either one with small pieces or another one with very long pieces. Which one easier to solve? Definitely the one with long pieces. So rather than solving 1,000-piece puzzle, it's easier to solve 10-piece puzzle. However, the long reads come with a cost, that is accuracy. So these long pieces might cause confusion or ambiguity when you try to solve it over here. So the short reads are still advantages. Also, their cost is cheaper than long read sequencing, but they are short. So they are a bit more difficult to solve than long read. And for that, which sequencing technology is the best? As I mentioned, there is always trade-off for everything. So there are short, but low error rate and cheaper. But here we have very long and high error rate and more expensive. There's a third technology from PacBio. That is a third company, different than Illumina and Oxford Nanopore. And this PacBio company provide HiFi reads, high fidelity reads, which is based on consensus. So we have the DNA molecule and we read it at least 30 times or at least 10 times. Third time provide very high accuracy, such that every time we read the same molecule again and again, we correct ourselves and we build a more accurate consensus or a read out of the same DNA strand. But the limitation is that as we repeat this process, the length of the DNA strand is always limited. We cannot go produce the same length as the read produced by Oxford Nanopore, for example. And for that, the HiFi reads length always limited up around 10 to 20,000 bases. But in long reads in Oxford Nanopore, we can go with 2 million or 2-point-something million as a maximum. The accuracy of the HiFi reads, although it's a bit long, but it's really doing well with the accuracy is really high, but it's also expensive compared to short reads as we have here. That is coming toward the end, and we see 3 main companies producing the read. Now, changing in these sequencing technologies over the time can definitely render some read mapping algorithm irrelevant. So those algorithms working for some other sequencing technology that is no longer used or it's not more advantageous, then of course this algorithm will not be used for the analysis, because all these analysis or all the downstream analysis used in genome analysis is based on the properties that we have for these genomic reads. For more information about how sequencing technologies dictate algorithm development, we have this very nice paper from our group where it shows the development over the last 3 decades and how some algorithms are still useful even in these days, and how some of the algorithms are not used anymore. So this is the last slide. So looking forward, if we're looking at 10 years later from now, will we still have a sequencing machine that give you the complete sequence as an output, or give you still chunks of your DNA? That's, we still don't know, but I think it's possible to do in the future, given the rapid advancement in all these sequencing machines. And even if we are reaching to this point to be able to read the entire DNA molecule as in single run or as single output, many of the algorithms used to do the analysis are still will still be useful in this case. Why is that? Because one fundamental step in read mapping or in genome analysis is to compare genomic sequences. And even if you have the complete genome, you still need algorithm to do complete genome comparison. So you need to compare genome to genome. That is still same concept fundamentally to comparing read to reference genome or read to read. But of course, it's more challenging since the search space is much larger than what we have in read to read, for example. So that is the end of the lecture for today. I hope you enjoy it, knowing more about sequencing technologies. In the next lectures, we will have more information about the computational steps done in the genome analysis and what kind of hardware acceleration can be done in all these. And we can discover new research areas together during the discussion. So thank you so much for watching. And thanks for all your questions. So we'll try to answer all of them in YouTube and Moodle. And thank you so much for now, and see you next week. Take care.